Okay, so this is the first in a series of videos about acid-base physiology. What we're mainly concerned with is the pH of your body. The normal pH is between 7.35 and 7.45. And remember, the pH equals the negative log of the hydrogen ion concentration. Remember, hydrogen ion essentially is representing acid. It comes from acid. And we measure hydrogen ion concentration on the level of nanomoles per liter. So this is the concentration we're talking about when we're at a physiologic pH, such that pH of 7.35 corresponds to hydrogen ion concentration of about 45 nanomoles per liter, and a pH of 7.45 corresponds to about 35 nanomoles per liter. So our body is going to fight to stay within this range. Outside this range, our proteins would denature, enzyme systems stop working as efficiently, receptors stop working, and this is when we get sick. So when the pH falls, let's say to a pH of 6.8, anything in this range we call an acidemia. And at a pH of 6.8, this represents about 160 nanomoles of hydrogen ions. Above this, we call this an alkalemia. At a pH of 7.8, this represents about 16 nanomoles per liter of hydrogen ion. The reason I put these limits here, 6.8 and 7.8, is this is probably the limit of at which life can exist. Beyond this, life is not possible. So the majority of the patients you're going to take care of are somewhere within this range right here. So how does your body actually stay within this normal, healthy range of pH? There are three main mechanisms. One is with chemical buffers. Two is ventilation for CO2 excretion via the lungs. And three, renal excretion of acid. And through this series, we're going to cover each of these mechanisms. So first, let's talk about all the chemical buffers. And the first most important one is the bicarbonate, carbonic anhydrase system. Okay, so this is an important equation. It shows the relationship between CO2, carbonic acid, and bicarbonate and hydrogen ions. I think whenever you're solving any acid-base problem or working in the ICU, I think it's worth committing this equation to memory. I think it will help you a lot. So here's what your body can do. It can take water and CO2 and convert it into carbonic acid. So this is usually a slow reaction, but in the presence of carbonic anhydrase, which is present in every cell, even red cells, it can happen a lot faster. So when carbonic acid is formed, very quickly it can dissociate into hydrogen ion and bicarbonate. This is how CO2 actually produces hydrogen ions and forms an acid. Likewise, this equation can proceed in the opposite direction. If hydrogen ion is available and someone adds bicarbonate to the solution, you're going to form more carbonic acid, which can be converted to CO2 and water. CO2, fortunately, can be excreted easily because this is an open system. Okay, but the main thing that we're interested in is the concentration of this, the hydrogen ion. And so how do we solve for this and thus calculate the pH? So I put on here the Henderson-Hasselbalch equation, which you may remember from your prior chemistry courses. pH equals the pKa of the weak acid plus the log of concentration of base over the acid. So in this case, the base would just be the concentration of the bicarbonate. Here, the acid is the concentration of carbonic acid. We can measure this, but we usually don't measure carbonic acid, but we can estimate it by the PCO2 because carbon dioxide is in equilibrium with carbonic acid, and only a small fraction of CO2 in the blood is actually in aqueous form. And so what we can do is estimate the PCO2 times the solubility factor, which is actually 0.03. This gives us an approximation of how much is actually dissolved in solution. So we can rewrite this equation. pH of this equals pKa, the pKa of carbonic acid, 6.1 plus the log of the base. So serum bicarbonate, the normal, I don't know if you know this, but it's about 24. And this is in mil equivalents per liter over the PCO2, which is about 40 torr, times this factor of 0.03. What this ends up being is about 6.1 plus 1.3. This ends up at 7.4, which is great. That's pretty much a normal pH. And that should make sense because I used a normal PCO2, right? And I used a normal bicarbonate here. And so I'm just trying to show you the relationship between the serum bicarbonate, the PCO2, and the pH. And this is actually how blood gases work. On a blood gas, the same one that you're going to order one day, we calculate, or we measure the pH, we measure the PCO2, and the serum bicarbonate is calculated. So that's how you get those numbers when you look at that readout. Okay, so now I want to talk about buffers. So do you remember that buffers in a solution, they function to resist change in the pH. That's in response to some sort of insult. I like to think of them as the shock absorbers. Any kind of insult that happens to your body. And the two main ones that we're going to talk about here is the addition of hydrogen ion and the addition of CO2. In those instances, a buffer is going to be something that's going to absorb that insult so that it actually doesn't affect the pH. Remember, our body really wants to stay within that normal range of pH. So here are two mechanisms for how we buffer these two types of insults. So here's your body. So imagine that we just added an acid. We'll call it HA. But remember that this is going to dissociate into A minus and H plus. So we've just added hydrogen ion to the solution. As long as this is free, remember based on what I just showed you, it could affect the pH. Here are some different ways we can buffer this hydrogen ion. There's intracellular mechanisms and there's some extracellular mechanisms. First, some of the intracellular mechanisms. Some of this hydrogen ion can actually be exchanged across the cell for other ions, mainly sodium and potassium. Usually these will come out of the cell in exchange for the hydrogen ion going into the cell via this antiporter. Once it's inside the cell, hydrogen ion can further be buffered against some proteins and even phosphate. As you can see, this is pulling it out of solution and putting it into the cells, which is very helpful. There's also these extracellular buffers on this side. Hydrogen ion can combine with bicarbonate, which is by far the most important buffer in the system. Do you remember? So look at our equation. Remember how this helps you? Hydrogen ion plus bicarbonate can form carbonic acid, in which in the presence of carbonic anhydrase, this could eventually lead to just the formation of water and CO2, which thankfully can be excreted in the lungs right here. To a lesser extent, some of the hydrogen ion can be absorbed by protein that's circulating. It can be buffered by some phosphate. Even bone does some buffering of hydrogen ion, but this is usually chronic in the setting of chronic metabolic acidosis. I'll just put a little footnote here. But for an acute change, like if we just dumped acid into the body, the bicarbonate would be the most important one in this extracellular compartment. And as you can see, as we added one hydrogen ion, we took away one bicarbonate in order to actually get rid of it. So that's why bicarbonate and hydrogen ions are always inverted compared to one another. Okay. The other type of insult to your body would be CO2, too much CO2 being produced. So what happens in response to this? Well, first, look at the top. Can extra CO2 be handled by our carbonic acid system? Actually, it would just form more carbonic acid, and that would form more hydrogen ions. So that's why this is dangerous. It produces an acidosis and will drop the pH, and that's not what we want. So these components can't be handled by this buffering system. It's going to produce more hydrogen ion. Thus, we have to find other ways to buffer CO2. One important way is actually intracellularly in red blood cells. CO2, remember, it can diffuse across the membrane. Once it's inside a red blood cell, CO2 can undergo this exact reaction because there is carbonic anhydrase in red blood cells, and this will eventually produce hydrogen ion and bicarbonate. And hemoglobin is actually a pretty good buffer for the hydrogen ions. And hemoglobin is also a good buffer for CO2 itself. I don't know if you remember this, but these effects called the Haldane effect and the Bohr effect, the fact that deoxygenated hemoglobin has a high affinity or a greater affinity for CO2 and hydrogen ions, but then once it obtains oxygen, it has a lower affinity for CO2 and it's able to release it. So, in the tissues where CO2 is high and oxygen is low, hemoglobin is available to sequester some of this CO2 and hydrogen ions to help buffer it. Likewise, on the other side here, this is how ventilation acts as a buffer. High levels of CO2 are sensed in the carotid bodies and the aortic arch, and via the vagus nerve, they tell the medulla to increase minute ventilation. And there's also direct sensors on the medulla itself. So in response to high PCO2, low pH, the medulla is going to tell your lungs to increase minute ventilation, breathe more deeply, breathe more frequently. And this is going to increase CO2 excretion. A lot of this can occur without you even knowing, so this is why I'm listing it here as a ventilatory buffer. This is how we can handle an excess load of CO2. So in response to your diet and end products of metabolism, there's going to be oxidation of amino acids like cysteine and methionine, hydrolysis of phosphoproteins, and then incomplete degradation of carbohydrates, fats, and proteins. And what this results in is a daily acid load, so production of hydrogen ions associated with what we're calling non-volatile acid. It can't be breathed off just like CO2, like we saw in the prior picture. This is just a consequence of being alive and eating. We estimate, and it's a good estimate, that about 1 mil equivalent per kilogram per day of this acid is produced. And imagine this amount being dumped into your body every day. And so that's coming in to the equation right here. And so look, on the other side of the equation, as a result of carbohydrate and fat metabolism, and here's the Krebs cycle. Remember, production of CO2 coming off the Krebs cycle, we will produce about 15,000 millimoles of CO2 a day. And we have to figure out a way to deal with it. This is termed volatile acid because CO2 is a gas, it's volatile, and we can breathe it off if we have a good open system. So every day, we have to handle all the carbon dioxide that's being produced, and we have to handle all the hydrogen ion that's being produced in order to maintain our pH in a physiologic range. Also, some of this bicarbonate here, remember, we want to keep that around. We want to keep the levels high enough to balance all the acid that we deal with. Some of this is lost in our stool. So we have to figure out a way to maintain the bicarbonate. We have to figure out a way to get rid of excess hydrogen ion, and we have to figure out a way to get rid of excess CO2. Fortunately, we have the lungs to handle all the CO2 that we need, because that's the primary job it plays in acid-base balance. So the lungs here will excrete CO2. On the other side, what about this non-volatile acid that we can't breathe off like we can CO2? For this, the kidneys play a critical role in excreting non-volatile acid. So the kidneys will do two things here. Another term for this is fixed acid, so I'll just call it fixed acid. And the other thing the kidneys do, remember the kidneys, they're filtering the blood, they're filtering everything. Bicarbonate is going to rush through the kidney. If the kidney wouldn't reclaim it or hold on to it, reabsorb that bicarbonate, it would spill out into the urine, and your bicarbonate levels will get low just because of that. So the kidney has to work hard at reabsorbing bicarbonate. So the second job of the kidney is to reclaim filtered bicarbonate. And so if the lungs are able to do this, and the kidney is able to get rid of this, hold on to this, everything will be just fine and you will have a normal pH. What we're going to go through next is how the kidney does this job. But I hope you can see if anything is wrong with any of these mechanisms, that's when you start moving in these directions.